Hello. In this lecture we are going to talk about macromolecules. The carbon atom has an important property that it can make 4 covalent bonds. So you can actually bind many carbon molecules, carbon atoms, one after the other and form long chains, molecular chains. And this is how macromolecules are formed, basically. Here you see a molecule with 8 carbon atoms forming a chain, and there is an OH hydroxyl group at the very end. The second important thing here is that these bonds, chemical bonds, the covalent bonds between the carbon atoms are sigma bonds. So they allow molecules to rotate along the axis of the sigma bonds. Gives the molecule not a rigid straight structure, but a more flexible twisting and turning type of structure. All these forms the molecule can assume at room temperature. All those three-dimensional structures you can obtain by twisting or by rotating all those sigma bonds is called conformers. The molecule can easily change from one conformer to another conformer at room temperature. There's no real barrier to this transition to occur. One simple type of macromolecule is fatty acids. Fatty acids are long carbon chains with which has a carboxylic acid group at one end. However, some fatty acids may have 1 or 2 double bonds in between the carbons. These double bonds actually fixes the structure of the fatty acid into conformations, which molecule cannot switch between these two conformations easily at room temperature. These are called isomers, and the name of these isomers are trans and cis isomers. As you can see here, cis isomer has a kink, and most of the naturally occurring acids, here the example is the oleic acid, which has an 18 carbon chain. Another type of group of macromolecule is the proteins. The proteins are polymers of amino acids. Amino acids, here you see a basic structure of amino acid. Amino acids are formed by a central carbon atom, which is called the alpha carbon. 2 groups are attached to the central carbon. One amine group, one carboxylic acid group, one hydrogen, and there is a 4th group, which is denoted R with a red R here, is a variable group. Actually, the amino acids gets its name from the identity of that R group. R group can be different things in different types of amino acids, but the rest is the same. So amino acids can bind to each other through a covalent bond called the peptide bond or the amide bond. This bond is formed between the carboxyl group of one amino acid and the amine group of the second amino acid, as shown here. And actually, this is a dipeptide, a peptide formed by 2 amino acids. You can elongate this dipeptide by adding or by binding more amino acids either to the amino end or the carboxylic end of this molecule and obtain long chains of peptides. Here you can see schematically the peptide chain. The blue is the nitrogen, and the red is the oxygen atom. And as you can see, the oxygen is double bonded to carbon. Carbons are shown in white, and gray boxes, light gray boxes, are the R groups. Is different for each amino acid. Oxygen double bonded to carbon is called the carbonyl group. So this chain here you will see as nitrogen, a carbonyl group, and an alpha carbon. This chain is called the backbone of the protein, or the backbone of the peptide chain. Peptide bond, even though it's a sigma bond, shows a quasi-double bond property, so it does not rotate. However, those two bonds, those two sigma bonds shown with blue arrows, can rotate. So this long chain of amino acids, the backbone, actually can twist and turn quite a bit, and this is called folding. Before going into that, I have to talk about another bond, which is neither covalent bond nor an ionic bond, which is called a hydrogen bond. It's formed between these two groups, the carbonyl group and the amine group of the NH group of the peptide bond. The hydrogen of the NH group has been bound to a very electronegative atom, a nitrogen, and the oxygen is double bonded to a carbon. Oxygen here is quite electronegative here. So this electronegative oxygen and the hydrogen, which lost its electron partially to nitrogen, the electronegative ion, can form a hydrogen bond shown with red stripes here. However, those two adjacent groups shown in the shown with red circles, they cannot form hydrogen bonds because their orientation is not correct. For a hydrogen bond to occur or to form, these two groups must approach each other at a certain angle, and there must be a certain distance between them. So let's continue with the backbone structure, keeping these in mind. Okay, here the backbone of a peptide chain is shown in green. Only the alpha carbons are shown with spheres. So you can see that because of those rotating sigma bonds, this long chain of amino acids, the backbone, twisted and turned quite a bit, and has a three-dimensional structure in it. And also you can see that there are multiple hydrogen bonds formed. Most of them are shown in red here, are between, as I've told before, NH and this carbonyl group of the backbone. But they are formed in between groups which are distant to each other. As the backbone twisted and turned, these distant groups come closer to each other at appropriate angles and can form hydrogen bonds, and many of them can be formed. There are also other types of hydrogen bonds formed, for example, between the side chains of the R groups of the amino acids, between different amino acids, shown in blue here. If the side chains has groups like OH or NH or COOH, can form hydrogen bonds easily. And also hydrogen bonds can be formed between the backbone or the peptide bond groups and the side chains of the amino acids. It's shown in yellow here. So with all those hydrogen bonds formed, this three-dimensional structure, this twisting and turning that form this three-dimensional structure, is kind of stabilized in that configuration. The thermal movement, the vibration of the atoms, may not be strong enough to break these bonds, all of them, and change this three-dimensional structure. So this structure is fixed by formation of multiple hydrogen bonds, which is called a conformation. Actually, this brings us to the three-dimensional structure of proteins. Proteins have four different structures. The first is the primary structure. It's called the primary structure. Primary structure actually is the sequence of amino acids. As we talked, a peptide chain is a group of amino acids bound to each other like pearls on a string of necklace. So the sequence of amino acids coming one after the other is the primary structure. Then the three-dimensional structures has two names. One is secondary structures, which are local but very strong structures. There are two types of secondary structures, the alpha helices and the beta sheets. These are relatively small structures, but they are very strong and quite stable. Then the overall three-dimensional structure of the peptide chain is called the tertiary structure. And if a functional protein requires more than one peptide chain to come together, for example, the hemoglobin molecule becomes functional if four different peptide chains come together and form the protein. So such proteins are said to have more than one subunit, and they have a quaternary structure, a fourth structure, which encompasses the overall structure of all the subunits. Here you can see a real protein. This is a three-dimensional crystallographic data showing all the atoms in the molecule, not the van der Waals volume of the atoms, but all the chemical bonds, the covalent bonds, and all the side chains are shown here. It's quite a complicated structure, but we can also show the backbone of the peptides. Only the alpha carbons are shown here. So alpha carbons are connected with straight lines or sticks. And each subunit has given a different color. So there are three different subunits here, three different peptides here that coming together and forming the protein. And these subunits, these peptides, are attached to each other through mostly hydrogen bonds. There are some hydrophobic bonds. There may be some ionic bonding, but mostly hydrogen bonds are responsible for the three-dimensional structure, secondary, tertiary, and quaternary structure of the protein. So hydrogen bonds are extremely important in the three-dimensional structure of proteins. You can also show backbone of the proteins as what is called the ribbon representation. This makes identification of secondary structures easier, and also gives a better idea about the general three-dimensional structure. Here you can see, indicated with the light blue arrow, an alpha helix looks like a spiral coil, and also the red arrow indicates a beta sheet, which is shown by flat arrows crossing each other. This is the van der Waals volume of the protein. So protein is not mostly empty. So as the other representations may suggest, that water or small molecules cannot penetrate into the protein easily unless there are channels or such certain structures that allows that. but of course the atoms occupy a volume in the space, and the protein is actually a solid structure. And also these the van der Waals sizes of these side chains also hinders certain rotations along the sigma bonds by bumping to each other, and also they contribute to the tertiary structure of the secondary and tertiary structure of the protein, which is called the steric hindrance, which limits the rotation around the bonds. This three-dimensional structure of molecules, which are called conformation, is very important for the function of the protein, as we will see in the next lecture. If this three-dimensional structure is disrupted somehow, for example by heating or by changing the pH to extreme values, can change or disrupt this three-dimensional structure, which is called denaturation of the protein, and then the protein loses its function. We will see why this is so important, why this three-dimensional structure is important for the function of the protein, but this is also tells us that the structure of this three-dimensional structure of these proteins is not the energy minimum of the system, because once you disrupt it, it does not fold back to its original configuration, which indicates that during synthesis of proteins, the folding has to be guided to a certain path. If the protein is left unguided, it may not fold properly. This is not the natural or the only state that the protein can fold into. So we'll talk about the function of proteins and kind of binding between proteins and small molecules in the next hour.